Hello everyone. In this video we're going to be talking about the pathology of Wilson's disease. So let's start. So what is Wilson's disease? So it is a genetic condition where there is an excess amount of copper in the body. This should not be confused with hemochromatosis, which is an excess of iron in the body. So in this disease, there are three main organs which are affected. These are the liver, brain and eye, specifically the cornea. We will see the details in just a little bit. Also, this is an autosomal recessive condition. Now let's see the pathology, but before that, we need to know what is ceruloplasmin and APOcelluloplasmin. So this is APOcelluloplasmin. Its function is to bind copper, precisely 6 ions of copper, which can then be used to transport copper to different parts of the body. Also note that whenever copper is bound to APOcelluloplasmin, it will no longer be called as APOcelluloplasmin, but now it will be called ceruloplasmin. Important to note. Now, what exactly happens in this condition? So we have the ATP7B gene, which is responsible for producing the ATP7B protein. So ATP7B is a copper transporting ATPase, and this is primarily located in the liver cells. Now this ATP7B has two functions. It helps in incorporating copper into the bile, and it also helps copper to attach to ceruloplasmin. And we just saw ceruloplasmin stores and carries copper from the liver to different body parts. Now let's see what happens in Wilson's disease. Here the ATP7B gene is defective. Due to this, the ATP7B is not formed, and due to this, the copper will no longer be incorporated into the bile or be able to attach to ceruloplasmin. Now what will happen? Since ATP7B is predominantly in the liver cells and now it is not working, this will lead to copper buildup inside the hepatocytes. And due to accumulation of copper and formation of free radicals, the hepatocytes will get injured. The copper, which is now in free form and not bound to ceruloplasmin, will leak into the blood, and this free copper which is released will accumulate in different tissues and will cause a lot of problems. So now finally coming to the clinical features. So here you are going to see a triad. Here the liver is going to be affected, the cornea and the brain. So what will it do in the liver? In the early stages, you will see that there is fatty changes in the liver, and in the late stages, it may progress to cirrhosis. Now what will happen to the brain? So mainly the basal ganglia is going to get affected, and this is going to lead to a lot of problems like psychosis, Parkinsonism, gait disturbances, poor coordination and even tremors. So these are important to remember because definitely in the question of Wilson's disease, they might mention any of these manifestations. And now what will happen in the eye? So there will be two things that you will be able to see. The first one is the Kaiser-Fleischer ring or KF ring, which is a ring of green-brown deposits of copper inside the Descemet's membrane. So everything in this line is very important to remember. Next, you can even see a sunflower cataract. So let's see how both of these look in a patient. So in this first picture, you can see the green-brown ring, which is the KF ring. And in the second picture, if you look closely, you can see a cataract which has a similar shape to a sunflower. Now, how are you going to diagnose this condition? So the levels of free copper will increase. And why is that? We just saw. Because copper is not able to bind to ceruloplasmin and so it accumulates inside the liver cells, breaks them and then escapes. Ceruloplasmin levels will decrease. Now why is that? Because what is ceruloplasmin? It is the copper bound form of APOcelluloplasmin, right? Now if copper is not able to bind, there will be no formation of ceruloplasmin. So hence the levels will decrease. And also the total copper levels will decrease. Now let's try to understand this part. So in our body we have mainly two forms of copper. We have the free copper and we have ceruloplasmin bound copper. But it is important to note that free copper makes up only 10% or less of the total copper in the body, whereas ceruloplasmin bound copper makes up to 90% of the total copper. Now both of these contribute to the total copper levels in the body. So it is very important to note that ceruloplasmin bound copper is the major contributor in the total copper levels of the body because it is 90%. Now what happens in Wilson's disease? So free copper levels increase in the body, right? We just saw that. And the ceruloplasmin levels decrease. So can you tell me what will happen to the total copper levels? Even though the level of free copper is increasing, but because celluloplasmin is a major contributor in the total copper levels and that is decreasing, so the total copper levels in the body will also decrease. So please don't get confused. This free copper that is increasing is the one that is going to accumulate in the different parts of the body and cause the manifestations, but the total copper levels will decrease. You can also do a urinary test to see the copper, which will be a screening test. You can also check the copper levels in dried liver, and if you get more than 250 micrograms, this will be confirmatory. And for this test, you use the rhodamine stain. Important to remember. And lastly, how are you going to treat this condition? So we can do copper chelation using D-penicillamine or trientine. Or we can use zinc-based drugs which block copper absorption from the gut. So that's it for this video. If you have any doubts, so please leave them in the comments. Or you can message me on Instagram anytime. I will be leaving the link for my Instagram in the bio. Thank you.